Okay, so thank you for the invitation. I'm Klara. I'm a PhD student in Metazoal Genomics Lab in Barcelona, under the supervision of Rosa Fernandez, and today I will present you the genome sequence of the Montseny horsehair worm, Gordionus montsenyensis, a key research to investigate Ecdysozoa evolution. Okay, so sorry. Nematomorpha, the animals that mold, include Ecdysozoa. Nematomorpha belongs to Ecdysozoa, which include Nematoda, Priapulida, Caenorhinha, Arthropoda, Tardigrada, Onychophora, and Loricifera. And besides Nematoda and Arthropoda, two phyla with high ecological and biomedical importance, the rest of the phyla are considerably understudied, with Nematomorpha, Caenorhinha, and Loricifera, not even one representative genome so far. The consequence of this is the poor resolution of Ecdysozoan relationship, and due to this, we don't even know where the phylum of Nematomorpha falls in the animal tree of life. So this high-quality genome of a nematomorph species will be a valuable resource and help us understand the evolutionary trajectory of the Ecdysozoa. So nematomorpha are parasitoid as juveniles, and they have free living stage as adults, and due to this peculiar lifestyle, they have lost respiratory, circulatory, and excretory systems. And so far, there are two orders described: Nectonematoidea, which parasitize mostly crustaceans, such as crabs and shrimps, and the freshwater or terrestrial ones, the Gordoidea, which utilize crickets, grasshoppers, beetles, etc., etc. So regarding the weird life cycle of these animals, the first peculiarity is that the only function of these animals is to mate. They don't even eat. The adults normally pair and mate in an aquatic environment and lay eggs, where and then the larvae, incised into within a variety of aquatic animals. These animals are the intermediate host, which is after being consumed by an arthropod, and this is the definitive host. Upon maturation, the nematomorphs alter the behavior of the host and manipulate them to jump into the water in order to make a successful transition from a terrestrial environment into the aquatic one. One could say that they create zombies that are seeking water sources. So we collected freshwater species, and apparently we collected an undescribed one that these morphological characteristics here indicate, and it belongs in the order of Gordioidea. It's freshwater, and in the genus of Gordionus. And we named it Gordionus montsenyensis after the location that we collected it, the Montseny mountain in Catalonia. So moving to the core of the project, we started with the sequencing and construction of the de novo genome assembly, which was part of the ERGA pilot project as one of the species of Spain. We generated long nanopore reads to acquire high contiguity, short Illumina data to improve the fidelity, and high C to reach chromosome level genome, and of course RNA-seq data for the genome annotation. From the genome profiling, we took an expected genome size around 280 megabase pairs, and that our species is diploid with not so much of heterozygosity. Regarding the assembly process, we did many tests and we followed different pipelines, and this is the final one. So we filtered the nanopore reads with field log, and then we used Ratatosk to error correct, since we know that nanopore are error prone. So we error corrected with the Illumina, and then we assembled using Flea. We paired the duplicates, we paired the assembly using the paired tubes, and then we scaffolded using the Hi-C and the integral software. As a last step, we did gap closing using the TGS gap closure, and this was our draft assembly. But we had some misassemblies, so we moved on with manual creation. This is the contact map before and after, and it's undoubtedly chromosome level, and the statistics corroborate this. We achieved an assembly of almost 290 megabase pairs, and scaffold N50 of 52 megabase pairs, and the L50 in the chromosome in the scaffold number 3, and the L90 in the scaffold number 6. And we have around 97% of the assembly anchored to 6 chromosome level scaffolds. From the cat plot, we have more than 95% k-mer completeness, which means that we assembled almost all of the raw genomic information that we had. But the peculiarities are the low GC content, which is one of the lowest in the animal kingdom, and this could be due to its parasitic lifestyle, and also the low Busco score that reaches 71.6, I think, complete plus fragmented, which could be due to the underrepresentation, most probably no representation of the this genome in the Busco, in the Metazoa database. Next, we annotated the genome assembly. First, the repetitive elements using repeat modeler and repeat masker. And then for the gene prediction, we used breaker 1 and breaker 2 with short and long evolutionary distance proteins. And afterwards, we combined the results using the Zebra software in order to get our final proteome. This pipeline yielded 63.45% of repetitive elements, and around 11,000 genes and around 12,000 transcripts. So the number of transcripts is low regarding to the total number of genes, which indicates that either our species doesn't do alternative splicing or we didn't capture it through our pipeline. And last but not least, more or less at the same time with our preprint release, 2 more genomes of nematomorphs appeared, which is great for the scientific community. But most importantly, we can see that our results are not a methodological artifact, because, for example, the low GC content and the low Busco score are 2 values that were deviating from what we expected. But no, it's just this enigmatic animal phylum. So I would like to thank Rosa, my supervisor, and all the members of Metazoa Phylogenomics lab. And we would like also to acknowledge FENAC for the sequencing, ARIMA for providing the reagents, and our source of funding.